Alors, bonjour à toutes et à tous. Mon nom est Louis Bernier, je suis professeur retraité à la faculté et toujours membre du Centre d'études de la forêt, où, comme diraient certains, je sévis toujours. Alors, aujourd'hui, il me fait plaisir de vous présenter la conférencière, la docteure Liina Jjürisoo, qui est maître de conférence à l'Université estonienne des sciences de la vie, située à Tartu. Et donc, le docteur Jjürisoo est de formation, elle est, et là, je viens de perdre mon nom, elle est, mon mot, elle est horticultrice de formation, elle a été formée à l'Université de Saint-Pétersbourg, à l'époque où Saint-Pétersbourg était encore en URSS. Alors, et elle a donc œuvré plusieurs années à titre de horticultrice, arboricultrice, et éventuellement a décidé de réorienter sa carrière. Elle est retournée aux études, cette fois-ci pour faire un doctorat, qu'elle a complété il y a à peine de cela 2 ou 3 ans. À l'occasion de son doctorat, j'allais dire qu'elle avait vu la lumière parce qu'elle a travaillé sur les ophiostomas, donc champignons qui causent la maladie hollandaise de l'homme. Et je vous le mentionne parce que c'est pas complètement innocent, parce que c'est lié à ce dont elle va nous parler aujourd'hui. Alors, présentement, Liina, en fait, est chercheur postdoctoral à l'université Linnaeus, dans le sud de la Suède, où elle travaille au sein de l'équipe de Johanna Witzel, dont les travaux, en fait, sur notamment le microbiome des ormes sont bien connus. Et dans le cas, alors, Liina est titulaire d'une bourse du gouvernement suédois pour une durée de 3 ans. Et dans le cadre de cette bourse-là, elle peut faire 2 stages à l'étranger. Alors, elle termine cette semaine un stage de 3 mois qui s'est déroulé vraiment vite ici à l'université. Et l'an prochain, elle va faire un autre stage de 3 mois chez des collègues de l'Université Polytechnique de Madrid, chez Juan Martin, le groupe de Juan Martin. Alors, tout ça dans le cadre de son stage postdoctoral. Alors, aujourd'hui, elle va nous... Est-ce que le titre n'est pas là? Donc, aujourd'hui, elle va nous parler, en fait, de la situation des ormes dans le nord de l'Europe et notamment dans la région des pays scandinaves et des pays baltes. Et alors, le titre est Situation of Elms in the Light of Dutch Elm Disease in Northern Europe. Donc, il me fait plaisir de vous présenter Liina. Alors, la conférence est également en ligne. So, for our guests who are participating online, from what I gathered from several countries, welcome to this presentation by our colleague, Dr. Liina Jjürisoo, who's in the final week of a 3-month stay at Laval University in Quebec City, where she's done a very, I would say, heavy load of work. As anybody who's visited our lab have seen the hundreds of petri dishes in the lab and apples in the lab can testify. So without further ado, I invite you, Liina, to give your presentation.
Oh, bonjour and merci. These are all the words I know in French, sorry. So my talk is going to be in English, as you understood. And I understood something that you told, but my first education was horticulture, then landscaping, then arboriculture. Then I defended my PhD on forest pathology and was lucky to get this postdoc position to Linnaeus University, which I'm very grateful for 3 years. Usually it lasts for 2 years only. And Quebec, or Laval University, is part of my project, but I will tell you about my project a little bit later. So my topic today is situation of elms in light of Dutch elm disease in the Northern Europe, as I had visited a lot of countries and collected thousands of samples. I think I have something to share with you. Talking about talking about elms, why they are so important, you have to know that in Europe the most, we have the three species, which is Ulmus glabra, Ulmus laevis and Ulmus minor, and all those are by distribution on their northern limit. Ulmus laevis, which is European white elm, is in the list of threatened species in most of the countries. But in Sweden, all those three species are in the red list. Elms are not so important trees for wood production, but they have very many cultural and ecological values. A lot of elms are growing in green spaces, historical parks, and that's why the mortality of elms is very important for people, because we are a lot of people living in the urban spaces, and they see what is going on, and they are they want to save the elms. So this is the thing that in historical parks, usually elms were taken like local ones. But since the end of the last century, they started to replace elms, so-called resistant ones or resistors. And they were planted also in this Northern Europe region. Yesterday I went to a concert, and like a so-called professional cretinism, you always see the elms. So there is some hope here in Quebec City, and also in anywhere else in the world, that we can save some of them or can have some of them if we still have some of them. Ulmus glabra is the main species in whole of the Europe. Overall, there are 0.2% of elms in the forests in Estonia, so I can tell about that. But they are spread scarcely, and mostly Ulmus laevis is only next to the rivers, but Ulmus glabra you can find everywhere. But they don't have the pure stands anymore. So you see, all the elms look fantastic. Doesn't matter, are they in the historical parks or in urban spaces, people love them, and they tolerate the conditions, urban conditions, and that's why they are especially precious for their value. But we don't want to see that kind of picture. It's made in southeast Estonia in 2018 when I was doing my PhD. So when you see that kind of picture, it's astonishing. We don't want to lose the elms, but we want to understand how to go further on. But do you know how beautiful is Ulmus laevis? You can see this fall color. This is totally different than regular elms. So you can see them bright yellow, or sometimes it's like looks like a violet. And this is very, very beautiful color during the fall time. And this is why Ulmus laevis is especially precious. So they have two different genotypes. Some of them get yellow, some of them get violet. And since 2015, they started to plant in Estonia resistor elms, mostly New Horizon. You can see next year after planting, the trees don't look so good. The top of the trees are dead, so they showed some dieback. But I collected samples, and no Dutch elm disease was there present. But those plants, the same elms were planted the same year also in my own town in Tartu. And in 2019, after 4 years, they look tremendous. So there is hope for urban spaces, definitely, but we can't plant them outside to the forests. So this is the main concern, that in forests we can't use them. We have to find some other solutions for that. So now going to the Dutch elm disease a little bit. Maybe most of you know the background of it, but it had been in the world more than a century. No good solutions how to fight this disease. And we know that there had been 2 pandemics. First pandemic was caused by Ophiostoma ulmi, which was less aggressive disease. And the second pandemic was caused by Ophiostoma novo-ulmi. So this Ophiostoma ulmi started in around 1910 and was killing about 10 to 40% of elms in Europe. But the second pandemic was much worse. It was taken to the American continent also, and it was much more aggressive, and only in UK, 18 to 90% of major elms in UK were killed because of that pathogen. Talking about the states, then the removing of elms, they had cost more than $100 million, and that having control over the disease, it needs the same amount of money. So it's a huge amount of money that that's needed to save elms in our urban space. And my question for you is, as I read and heard about that, Alberta and British Columbia, it's like free of Dutch elm disease. This is a question mark. After I'm finishing my presentation, I will come to this question again. So you never know. If it's not recorded, it doesn't mean it's not there. How the symptoms are looking like. Usually people are looking at the trees. Okay, there are some yellowing parts, but does it matter? Sometimes some twigs are dying and no problem. But in 2 weeks, or even maybe in 1 week, you can see the so-called flagging, and it is a concern. Then you know definitely Dutch elm disease is present. And when you take the twigs which are affected by Dutch elm disease, you can see the rings in the twigs. It's caused by tyllosis, and it's definitely that it's called CODIT. It's compartmentalization of decay in trees. And trees are starting to protect themselves, starting to act against the fungi, but at the same time they are killing themselves. They don't want the disease to spread, but the disease spreads quicker than the tree can act against it. So those metabolites, they block all the functional vessels, and that's why the trees are going to die very quickly. In 1 or 2 seasons, the huge major elms are dead because of that. Always we are talking about how the disease is spreading. So there are vectors. So in American continent, there are that type of beetles. Scolytus multistriatus is also present in Europe, but also it's spread by root grafts, and international trades somehow is also something to do with that, because first time when the disease was brought over the ocean to the America, it was brought here by logs. So the trade is not the best option. So they always show, connected with Dutch elm disease, that kind of scheme. But I also was happy to make some pictures by myself. So I met Scolytuses by myself. So you can see they're tiny, tiny beetles. And when you're in the right time, in the right place, then you can see how thousands of them are moving on the bark. You can see the entry holes, and next year the picture will be like we see on the right. So they're called bark beetles larva patterns. So vectors of this pathogen in Estonia, I found species like Scolytus, multistriatus, triarmatus, and laevis. And by the way, multistriatus and triarmatus are very similar. So only experts can say which one. They have to find the genitals and whatever they are taking off the beetles to understand which kind of species it is. So they're very, very, very similar. In St. Petersburg, I found only 3 species, but the most we have to worry about this Scolytus pygmaeus, which is only 2 millimeter bark beetle, but it was brought together with nursery trees to St. Petersburg. And there are thousands of them on the trunk, and they are killing the tree already because they are present. So I was happy to do some sampling on beetles, so I collected by my handmade trap, and there is a pheromone inside this black plastic, and it comes from, by the way, from Canada. So I ordered it from Canada, and I filled the lower bottle with alcohol. And what happened? Not only bark beetles were caught by these traps, but we also got some other different species. But the most interesting species that I found then was that 2 species, Xyloborinus successani and Xyloborus dispar, they also had Dutch elm disease on their skeleton. So it was like you never know. You can't say that there is only Scolytus spreading or specific bark beetles. There can be another species as well. And when we collected something in Norway, we found also under the bark some other beetles. So talking about the pathogens, as I'm a pathogenist in Estonia, it was reported only in EPO, which is, it's called the European and Mediterranean Plant Protection Organization, where all the diseases are registered. So in this list for Estonia, it was Dutch elm disease was registered in 1979 only. But talking about when it was identified in Estonia, it was 2006. Just the main species, but in 2017 I found out that we have also both subspecies of Ophiostoma novo-ulmi. Talking about Russia, then Dutch elm disease was there present already, or registered already in 1939. And reported in Nepalis as well as in Estonia as Ophiostoma ulmi in the same year. So why Russia is for me important? Because I studied there. I graduated St. Petersburg Agricultural University, and as Russia has border with Estonia, then the diseases doesn't know borders, as you know, and there is not so much access for other people to go to Russia. So I used my position and my friends and everybody, and knowing the language, it was easy to go and collect something, especially when my friends are also from the horticulture, so it was easy. And I liked St. Petersburg a lot. Talking about elm disease in Nordic countries, there had been surveys done by local scientists, and a lot of information came out in 2016. But in Finland and Norway, this disease is not registered until now. So this is interesting, especially for us, is interesting because they both are next to Russia as well. So I will show it further on. We also wanted to understand, does the different subspecies affect the rate of death for the trees, and also the health condition for the trees. So we started to register the health condition of certain trees, and we found out that Ulmus glabra had always the worse health condition than Ulmus laevis or those hybrid elms. As in Estonia and also in Russia, you can see. So the red one is the healthiest one. The blue is a little bit affected, but not definitely by Elm disease. But there can be other reasons as well. So you can see that definitely Ulmus laevis is in the better condition. Talking about the background of Russia, coming back, the situation there worsened substantially in 1995 when they started to register everything. So you can see how many plots in 2002 were. So it means plots, parks or alleys. And in 2015 there was already 700. And in 2015, just more than 5,000 dead elm trees were cut down, but now it can be already hundreds of thousands. And in Russia, they used a lot of kilometers of alleys, just pure elm stands, and you can see how they are dying. The picture was very, very awful. So, to compare the vitality of elms affected by the two subspecies, Ophiostoma novoulmi and subspecies americana and subspecies novoulmi, we found out that in Estonia, in our capital, there was subspecies americana, and the certain trees we put rated their health, and also in South Estonia, we rated the trees after 24 months again, and we found out that in Tallinn region, subspecies americana caused quicker death for trees than in the conditions of subspecies novoulmi. And it was based only on Ulmus glabra. So the sampling sites in Russia, to show some maps as well, you can see this is the pathway from St. Petersburg towards Moscow, and these the last flags are on the border with Finland, and that's why it was most interesting thing to understand, did the disease hop over from Viipuri to Finland as well. And here is our border with Russia. So 300 kilometers, sometimes it means can be a long distance, but you never know. So in what happened in St. Petersburg, they got the plants long alley. They bought they knew that they bought resistant elms called resistor, but they were cheated, and some local businessmen changed the trees and bought some cheaper ones and just changed the variety. And this variety that we found on this alley was dodoens, which is not, it's quite susceptible to the disease. But they didn't know that they could check if the tree is resistant one, because the nurseries are putting the chips into the trunks, and you can check it out. Is it the real one or not? To be sure that, because not only the specialist understands what the species or what the variety is. So it was a very, very sad story, and together with those trees, they also bought this Scolytus pygmaeus. So it was like a double problem for the town. So how I found out that we have subspecies? So first of all, we always have to isolate the fungi from the twigs, because there is no species-specific primers for Ophiostoma novo-ulmi. And detection of the pathogen from pure fungal cultures by ITS region, it always gives us an answer, just Ophiostoma novo-ulmi, no subspecies. So I confirmed always my findings by sequencing the PCR products. But identification of subspecies were done by specific call region. And here we can find difference in only 6 nucleotides between those two subspecies. They are very, very similar, but somehow I think that subspecies americana, at least on our continent, is a bit more aggressive. And to find out if there are hybrids between those subspecies, we also had to do something with CU gene region, and we amplified this CU gene region with restriction enzymes and found out that kind of picture, which was made by putting them into agarose gels under UV light. And the picture is, you can see that the novo-ulmi gives a higher amount of base pairs, and Americana gives a lower amount. So this is how I found out that based on those two genes, call region and CU region, we can have hybrids, but we don't know what's going on the other genes. So this is why we here want to understand and send the samples to some other sequencing to understand if there is some more hybridization in those pathogens. This picture I like most. This is a probable distribution of elm disease agents in Estonia, made by my colleague. So the first foundings of Nova Ulmi and subspecies americana, it's in the different places. And this distribution map shows the locations of specific infected trees. You can see the N or A. And they were, again, identified molecularly. And the distribution area of Stoma americana is here, black dotted, and the Nova Ulmi is colored by the years, different colors. It should be noted that this distribution map does not show earlier findings of this pathogen, only since 2013. And the thing is that the actual origin and distribution centers of the pathogen, Dutch elm disease and its subspecies in Estonia can be identified anywhere else, and this map is based on 2020, but I have collected samples also all the other years, so I know now that this is not the true map right for now. I have found several hybrids and several locations where Americana is present. But what is interesting based on this map, you can see where the Americana distribution area ends, and just on the line you can see this small X, and it was the first finding of the hybrid in Estonia. It's like just on the line where the two subspecies met. So this was interesting for me, this kind of finding. So in Russia we found all mixed up pure subspecies and hybrids, and then I wanted to understand which is more aggressive of them, and I decided to do some fungal growth rate measurements. It was very elementary and primitive. Nowadays I would do something else, but you can see as the pure subspecies are almost with the same growth rate, and the hybrids are growing much more quicker. It doesn't mean maybe that they are more aggressive, but it can mean that. So, as already Professor Louis Bernier mentioned, that we did a huge new trial for understanding what the growth rates is doing with different samples, and we definitely made 960 plates for this trial, and we did 3 repetitions for all different pure cultures and put them into 2 different temperatures. We have the results already in our Excel table, but we had no time yet to find out what is going on. So it's very interesting what we are going to find. So you can see even the picture of the pure cultures can be very different. They can vary. There are only 3 examples that I put here, but there were 9 different groups but we could do also subgroups with them, so it was interesting. So I made photos and we measured them, and we will find out, is there any correlation between aggressiveness and growth rate, maybe in the future. Then the next thing that we did some inoculation tests on apples, which is more human, maybe humanistic, than killing trees. Because apples are anyway going, if not through our guts, then they degrade one day. But we wanted to understand how the necrosis rates are showing on different pure cultures. So 300, almost 300 apples. And today even we went to measure the apples, because we have to measure them after 14 days after inoculation and 28 days, which is going on. And it's not so easy that you are just taking apples. You have to wait until they are quite yellow. So then you can only inoculate them. So it's very interesting what is going on, and can we compare the aggressiveness? It's very interesting results that we are waiting for. So, first of all, there is a question among the scientists that, do we want to understand where this disease started from? Because Dutch elm disease, it doesn't mean that it started from Holland. It was just described by Holland scientists. But it was thought that it started from Ukraine or somewhere there. And I was lucky to collect some samples also from Ukraine, so maybe in the future we will understand more if we are going to do some population genetics based on that. But what I wanted to show you here on this map, so it's Estonia is it was. Why it's not going? Okay. Can you see? No. Okay. It's all the map is in Estonian, so I wanted to show you. So you see where is Tallinn, where is Helsinki, and where is Sweden and Norway. So I was lucky to collect samples in 2022 and also previous year, together with some colleagues in Sweden. This year I was lucky to go to Norway and collect some samples, and the last year I went to collect some samples from Finland. So now we are going to register officially that there is a finding in Finland already, in Helsinki, and it was subspecies americana that I found. So to zoom in a little bit, I'm showing you how many samples I collected in Estonia. So it's almost covered everywhere. Every time when I find something, I remember when I was doing my PhD, my children were together with me, and when I showed them, oh, that is a dying elm. No, Mom, leave us alone. So it was like, for them, it was like a nightmare, but it was like my passion to collect them, to find them. So almost all Estonia is covered. And by the way, those stars, they are from findings from 1939. And what is interesting for me, then only 2018 in some regions, this Dutch elm disease started to occur again. I want to understand, is there any connections between those previous findings and that then it escaped or went underground? We don't know. And why now again those diseases, this disease is up again in the same region? So this is, for me, biggest question, concern connected with Estonia. So zooming into this Scandinavia, Nordic countries, I'm showing you a small part of Gotland Island, of Sweden. The samples were collected by my colleague, but you can see there are all possible varieties. Americana, and then the green one is Novo-Ulmi, and also 2 different hybrids, and they are all close to a harbor. So we have to think about it. So this Scotland is most investigated region for Dutch elm disease in Sweden. And the Ulmus minor is growing mainly on this island, not on the other spaces in Sweden. So to zoom in this region where my university, Linnaeus University, is situated, you can see also the same. We can find both subspecies and also the hybrids between them. So it means that they must be met long time ago. But what happened with Norway, where I was this year, we collected a lot of samples, and next to Oslo, there were hybrids we found, and also hybrids on the south coast, where this disease had been present a longer period. But what we found in this fjord area, there were only pure either Americana or Novo-Ulmi. How this disease occurred in the middle of nowhere, nobody knows. Maybe future will give us answers. But first, when we were driving by boat to look at the situation on this fjord, and you can see marked with this yellow arrow, there is a dead elm, and further on you can see as well, and the slopes are so high you can't do anything with the trees there, because you can't even climb there. And the situation came worse and worse when we came deeper into this fjord, where the fjord ended, and you can see dying elms, and this situation is probably the most horrible situation I've seen connected with elms. So those areas have always had broad-leaved forests, and if you can see there are Scots pine. This is all planted, and the Scots pine has their own problems, like in everywhere in Europe. But there had never been conifers in this region before. And also this nice and beautiful in one village called Leikanger, the arborist came to help us and climbed on the tree, and he wanted to understand, shall he take it off or not? So he took the twig and dropped it down, and the answer was yes. So he had to take it off even though it was the most beautiful tree in the region. And now talking about my project, it's called New Approaches for Conservation of Threatened Elms, Elm Trees through Holobiont Concept. What does it mean? It means that it acknowledges the potential role of the associated microbiome as a potent factor behind the tree's resistance to pathogens. So there is not just Dutch elm disease, there are plenty of some other microbiological things that are inside the tree and we don't know yet what is going on there. But the overall goal of this project is to expand the current knowledge base for development of sustainable conservation strategies for elms under this threat of this disease, Dutch elm disease. And in Sweden, there is also a national effort to develop long-term strategy for conservation of the trees. But for that case, we have to find their survivals. It means in Sweden, now people can register their big elm trees which are surviving via one link, and then my colleague is taking contact with them and going to visit those trees and find out are they really the survivals. And sometimes they are not so survivals because, as on my first slides I showed that the Dutch elm disease starts from the very up from the crown, so people don't even maybe realize that disease is present already, so it has to be checked. And so we are trying to find out the potential connections between Dutch elm disease resistance and composition of the fungal communities in elms. Soon my next article is submitted based on that. So I collected twigs from different areas. They were, some of them were healthy and some of them affected. And by high turbulence sequencing, which was done in Norway, we are finding out which is the fungal composition inside the twigs to understand is there any correlation. There are some, but it's not so easy to find. So there are so many fungi that we can't isolate even, which pop up from those high turbulence sequencing. So also we want to understand what does the vaccination do for the trees, so then composition of fungi inside the twigs. So vaccination done in Europe is usually based by Dutch twig, which is verticillium strain put into the trunk, and it has to be done before the beetles start to fly, and it only helps to improve the immune system of a tree. It doesn't kill the Dutch elm disease fungi. So you have to be very, very precise timing. This year, what happened in Europe, spring came 1 month earlier, and the vaccine was not present yet when the beetles started to fly already. And we saw in Norway that it affected a lot the vaccine trees, because it wasn't in the right time. But I did a small trial also in Estonia, so I collected samples before vaccination and 1 month after vaccination, and now we are waiting for those data coming out, and also an article will follow based on that as well. So resistance tests are very important, and this is the thing that I'm going to do my next 3 months when I'm going to spend in Spain. and this will be the next trial for me. But we are using the data we found out here in Quebec, so it's like a prolongation of everything that we are finding out here, and those visits for those collaborators organizations are very precious for me, because 3 months being with Professor Louis Bernier, it's like it was my dream when. I was on the conference once and asked him, can I come and visit your lab? Of course, if you find money. So here I am. So it was so precious for me. We also are doing still cooperation with the Swedish University of Agricultural Sciences, with other scientists, not to do everybody in their own corner. So we decided to gather all together and solve the problem in the whole Sweden all together, maybe for the other countries as well. We are also doing cooperation with the Skogforskning, which means Forest Research Institute, and definitely with University of Laval and Technical University of Madrid, where I'm going next year. So it's very precious for me. And my supervisor for my postdoc is Professor Johanna Witzel from Linnaeus University. So she asked me, are you ready to live in Sweden? Yes, why not? My children are adult ones, so I can go anywhere. Now I'm free to go. So, to show some sad pictures, there was always thought that Camperdowny is not affected by Dutch elm disease, but this picture is made by me, and you can see this sad situation in Estonia, and I saw also in Sweden as well. Also, sad situation when I have previously told you that Ulmus laevis is less affected. The reason why it is less affected is one thing: the beetles don't want it. And the other thing is that it has narrower vessels. But it depends also on where this population comes from. So here you can see 64 trees in St. Petersburg, Ulmus laevis, killed by Dutch elm disease, and also you can see beetles on the bark. But I realized that the leaves and all the composition of those trees were totally different. The seeds and flowers were like Ulmus laevis, but it was very hairy for like Ulmus glabra. But they never get hybridized because they are blooming in the different times. So it depends also the genome of the tree, how resistant they are. So my question is there any hope? If we look at this tree, it's a tremendous tree. I'm admiring at this tree every year in Tallinn. There had been at least 50 Ulmus glabras on this row. All the other trees are dead. Definitely this tree had a root connection, root grafting between other neighboring trees. But this is standing so nice, so green, and maybe this genetical background is something that we have to understand why it's not affected by Dutch elm disease. So this is the best thing. And I have seen, of course, in some regions where Dutch elm disease is present, there are trees, the survivals. So my answer is yes. And coming back to the question about Alberta and Canada, based on that. There was nothing in Finland, there was nothing in Norway, but now it is present. So if it's not registered, it doesn't mean. And when in Norway, when we were in this region and asked the local people how long this disease had been present, then people don't even understand how long it had been. Some say that some 10 years, some 20 years. So the disease had been present, but the scientists hadn't been present yet. So the knowledge about that the disease is present we got from arborists. So this is very precious for us to be connected with arborists. And as I'm an arborist, my background, so I'm also part of ISA. I leaded my Estonian Arboricultural Society as an associated member to ISA. So that's why I was very closely connected. Not anymore because I had to defend my PhD. So you know how long the time it needs. And this is my final slide. So, if you have any questions, you're welcome.
Thank you, Liina. And if there are questions, just raise your hand and I'll give you the mic so people who are online can hear you.
Okay, thanks, Louis, and thanks, Liina, for that wonderful presentation. I'm very intrigued by the hybrids that you were showing earlier on the slide. And you're looking at whether those hybrid fungi is more aggressive than the ones we expect to harm the elms that we are seeing. But my question is, wouldn't the hybrids are potentially infertile? Wouldn't we expect, and this is a hypothesis obviously, wouldn't we expect it to stabilize at some point? Are they able to, like, how do we see those patterns emerging in the future?
Thank you for the question, but I think that the hybridization right now started not so long time ago. So it takes time when it starts to get even. This is the question that we want to understand while we did our trials here, to get something, some connection maybe. So we don't know yet, but maybe in few months, few years, we will know more about that.
That would be really exciting because you would expect them to be aggressive right now, but eventually that might also cause a diminution in infections, right? I don't know. We don't know.
I'm not that good expert on this diminishing, but I think Professor Louis Bernier knows about that much more, and he is doing some experiments on some things.
There, I'm put on the spot. Well, I'll base my answer on mostly, I would say, on Clive Brazier's work, because he was the first one, in fact, to describe the new species of Novolmi, and then to describe subspecies within Ophiostoma novolmi, and then to describe the emergence of hybrids between the subspecies. I mean, there are hybrids between Ophiostoma olmi and Ophiostoma novolmi, but most of these hybrids, they don't have a good fitness. So, and in fact, Ophiostoma olmi not only is less aggressive, it is less fit, it has basically pretty much disappeared. It should be on the red list of endangered species, but no. Although there's still some Olmi DNA around that has been acquired by Novolmi, but within Ophiostoma novolmi there are hybrids between the two races, subspecies. Sorry, I show my age. It used to be called. And Clive Brazier has described what we call like hybrid swarms, which is a phenomenon that's well known. I guess Pauline could tell us about that because that's something that interests her. But those hybrid swarms are basically populations of hybrids, and their fitness tends to be higher in that case, and they tend to be more virulent or more aggressive. So far, this is what has been documented in terms of hybridization. When we see the also been cases of like strains where the aggressiveness goes down. In that case, sometimes we have other phenomena like the presence of viruses that attack fungi, and then that tend to and in fact, people who work, for example, on chestnut blight know that phenomenon very well, and in fact, it has been used to come up with and deploy like a phenomenon known as hypervirulence and as for biological control, but I'm going far.
Okay, thank you.
Thank you, Liina, for your presentation. For reference, there is the Elm disease in Alberta. It has been recorded in Lethbridge in 2022, and we have samples if you're interested.
Okay.
I wanted to know, what do you think about our management strategy regarding the Elm disease? If we cut down trees as soon as they are infected, we also prevent ourselves from finding resistant trees. So we limit the spread, but we also potentially cut resistant trees. So what would be best?
Yeah. This is a good question because based on that I've seen on Ulmus laevis, they are also survivals even though they are affected. The trees will survive even if the disease is present. I've got the samples from them, but I'm going next year and the next year, and they are still alive. So this is also a thing that to think about it. And when we are cutting very soon, anyway, the beetles, you never know where they are flying and how they are flying, and is everything covered, and where we are taking them. And one good explanation for how the beetles can spread, that usually they say they can fly 40 meters, somebody said 400 meters, but there was a case in Estonia where in the middle of nowhere, in the middle of the fields, there was an arboretum, and after a huge hurricane, next year there were spruce bark beetles and Dutch elm disease present. So maybe the winds can also bring those bark beetles to us. Nobody had proved that the spores are somewhere. But I'm thinking that it has to be also an option, that the spores are spread, or they are somewhere under the ground. You never know, the nature is magnificent. So yes, but in urban space, we have to cut them down, otherwise people are very worried. But in the forests, you never cut them back. So maybe this option in Norway, where we saw the ugliest picture, maybe some survivals are there, but somebody has to climb and find them out there.
So we should look for resistance in natural populations.
Yeah, that's it. In natural populations, I think it's even no reason to cut them down, because they anyway have the root connection between each other. And I've seen that also Ulmus laevis died if it had a root connection with Ulmus glabra. And a little bit later, but still. So this is not so easy. We don't understand nature at all.
Hi again. I just want to add a comment, and I want to also ask for your perspective. So Allison and I, we were just in Winnipeg, a couple, and Louis too, just last month. And Winnipeg, I don't know if you know the story, they have a hold on the Dutch elm disease, and about 60% of their canopy in Winnipeg is elms and ashes, that two species that we are scared to plant here. And a lot of them in Winnipeg, what they were sharing with us is they were, a lot of them were resistant varieties that they were planted, and they are able to withstand the stressful conditions that cities you find yourselves in. And my question again, coming back to Pauline's question, yeah, like we do cut them down. That's one of our like #1 management strategies. And in terms of trees, okay, yes, we have elm, like genes of, genotypes of elms in the natural forest, but in cities, it's much, like, the fitness is much more, probably more enhanced because of the adaptations that they have to go through, either by humans and by environment itself. And if we cut them down, like, how are we able to find strains that are much more resistant over time? I mean, is there another way that we could do this without infiltrating the natural forest?
And by the way, comment to your comment, I started to collect also the buds and leaves from different trees. So I have them thousands stored in our freezer. And I'm waiting the right time to start to do something, the DNA of trees. So now we know which ones are dead, which ones are survivals, and to compare this genetical background. This is the thing. And the thing, even if we are finding the survivals, nobody ever propagated Ulmus glabra by cuttings. Everybody did it by seeds because it was so easy. But if we are finding the resistant trees, then we have to start to think how to propagate them. It can't be just one tree. There has to be thousands of them because the variety has to be also, we can't do it based on just one tree, so it is more complicated, all those things. So I want to work out also the propagation, as horticulture is in my first love. I like propagate everything. So propagation almost glabra by something else. Okay, in vitro is of course one option, but are there any other options for that as well? I don't know. Ulmus laevis, everybody knows how to propagate it, also from cuttings, and Ulmus minor, it gives even root sacs, so it's easy to do it, but nobody is propagating them anymore. So if we even want to do it, to give it back to the forest, and by the way, for instance, in Estonia, foresters already want the broadleaf trees back. They want to do mixed stands, not anymore just pure stands, because you know what is happening when there are pure stands. So people are more interested in that as well.
May I ask a question? Liina, I have a question for you. You know, there are some tree diseases that are very specific. For example, poplar rust attacks poplars. On the other hand, you have verticillium wilt, which is a disease very similar to Dutch elm disease that attacks over 100 different plant species, including elms. What about Dutch elm disease? Do you think, should we consider Dutch elm disease to be a disease specific to elms or not?
Me personally, I found Dutch elm disease also on Corylus avellana. So, and I also read from the articles that it had been found on Fraxinus species. And we know that when we registered some pathogens from nursery trees, it was found on Tuja. So you never know what is going on, because if there are no elms, the beetles want to live. So they will find something. And all those microorganisms as well. Either they are dying or they are finding something else. So personally, I found on Corylus avellana, so it can be everywhere.
But and you saw symptoms on Corylus?
Yeah, they were symptoms, and I isolated from Corylus avellana. And
yeah, because that would be detrimental here. We plant a lot of tuyas, and Fraxinus we already deal with that. The tuya that would be.
It's just by high turbulence sequencing, so first results that we found. So you never know where you can find them.
And you mentioned that when you were doing your surveys of beetles on the bark of elms, in addition to these colitis, you also found xylo something?
Xyloporinus,
Okay.
yeah.
Do you know which species these insects are normally associated with? Are they generalists?
Generalists, yeah.
So if they carry spores of Ophiostoma, that means they can...
That's it.
propagate them.
They are usually feeding on fungi. So there are no questions.
No, there are no questions. So, which means, Janani, you have the opportunity to ask a question.
This is my last question, by the way. I'm just surprised that you found it on Tuya. Is it Tuya occidentalis? Okay, so was it a hybrid fungi or was it just a...
You can't say by ITS region.
Okay.
The high durability sequencing is done by ITS region,
Okay.
so you can't do it. Right now, we don't have those PCR specific primers.
So you detected it just on one individual or a number of different individuals?
No answer, because I don't know yet anything.
Okay, it's not
just
conclusive.
We just got the results lately.
Okay, this is interesting to follow.
It may be also, it could be also be in the soil, because we took also samples from soils, so it definitely didn't have to be on the tree itself. So I will let you know.
It's my last one too. Is there any hope? Yes, and I would add, are we close to getting answers? I think yes, too. I think by combining our extensive North American sampling, your extensive European sampling, adding sequencing, we can really get a much better picture of what is going on, doing some demographic inference to understand how it happened, because as you underline, like in a region in Estonia, there was a first wave and then it just died out and then it's coming back again.
Mm-hmm.
Like it's in North America, it just popped out of nowhere, like around the Great Lake regions, but we don't know why. But I think we're getting closer and we will have some answers.
We will do our best.
Yeah.
I think in this case, cooperation is the most important thing, because otherwise we don't find the answers. So that's why I like to be everywhere and cooperate with people, because it's so impressive for me.
Dernière chance pour celles ou ceux qui aimeraient poser une question, qui n'ont pas encore eu l'occasion, parce que c'est toujours les mêmes qui posent des questions. Alors, j'aimerais vous remercier de votre présence. J'aimerais remercier donc Liina de sa présentation et aussi pour tout le travail qu'elle a accompli au laboratoire. C'était vraiment très agréable de l'avoir parmi nous. Et il va faire un peu plus chaud en Espagne quand elle sera là, mais elle était très contente de voir la neige. Donc, il y aura sûrement d'autres conférences. Restez à l'écoute. Là-dessus, je vous souhaite une bonne fin de journée. Et printemps, surveillez les ormes.
Merci.